And now for our presentation, Dr. Abhijay Borole will join me to provide you with an overview of a new system for microbial electrolysis for renewable hydrogen production. Dr. Borole is a research scientist in the Biosciences Division at Oak Ridge National Lab. He is a chemical engineer with 16 years of experience in energy research and development, including renewable energy. That includes biofuel, biogas, bioelectricity, and fossil energy. His interests lie at the interface of biology and engineering and are multidisciplinary with expertise in biocatalysis, electrochemical, and biochemical engineering. It's my pleasure to introduce him today.
Thank you, Nestor. And I would like to thank our Technology Transfer Department to allow us and provide this facility to communicate our research to the outside world. So today I'm going to talk about microbial electrolysis and hydrogen production. So here's an outline of my talk. I would like to poise the problem that we are going to discuss today with the emerging bioeconomy and how it fits into the future of the 21st century. I will then discuss the microbial electrolysis technology and the advancements ORNL has made in this field, and then discuss in depth the research we have done in the area of biorefinery and discuss its implications for application in the field, as well as touch upon briefly on the oil and gas industry applications. The emerging bioeconomy can be defined as the economic, environmental, social activities associated with the production, harvest, transport, conversion, and use of biomass as a resource for producing power, bioproducts, as well as biofuels. There are a number of other definitions that have come up in the literature, and I have added a couple of them down here. Essentially, the future economy will include biomass conversion to various products as a main driver for the economy. So in this slide, I would like to introduce you to the biofuel production process in biorefinery. In this simplistic diagram of the biorefinery, it shows conversion of biomass to biofuels. And it includes a pretreatment step, followed by fermentation, separation of the biofuel from the stream, and ending up with a product which is called as distillation bottoms, which essentially includes unconverted biomass as well as lignin, which is then typically used to produce additional energy via combustion and other thermochemical processes. It also results in production of wastewater and additional electricity potentially from burning lignin. The energy efficiency of this process, as it stands, it's about 53.5%. So going into a little bit more deeper into this process in this diagram, which is a little busy, however, I would like to point your attention to, in addition to the energy efficiency, which results in losses of about 46%, there are also losses of water in this process, primarily due to use of evaporation, boiler technologies, and so on, which result in evaporation and so other means of loss of water, so which can amount to about 95%. So in addition to the losses that I pointed out in this slide, the composition of the wastewater that is generated in the biorefinery is also pretty complex, and it has some components which are inhibitory to the overall process if you have to consider water recycle. During the biorefinery conversion process, in the pretreatment step, the processing of biomass results in production of some molecules such as furfural, hydroxymethyl furfural, lignin degradation products such as phenolics, as well as acetate from the hemicellulose. Some of these molecules are shown here. So the wastewater that is generated in the biorefinery is a complex mixture of these kind of molecules. Some of these molecules have been known to be inhibitory to the fermentation organisms that are typically used to produce biofuels. When you want to produce biofuels and in order to make that process economic, you have to treat the biomass at high loading conditions, which exacerbates the problem of these inhibitory contaminants and making the water recycle almost impossible. So if you look at the treatment cost for the wastewater in the biorefinery, it has been reported recently that it can cost as much as 34 cents per gallon of biofuel produced, which amounts to about 15.8% of the total cost of producing biofuels. So given these problems, we would like to address and think about what alternatives are available to handle the waste. So there are two primary streams. One is the biorefinery process water, which is generated after fuel recovery, which can contain anywhere from 5 to 15% of the original biomass energy that comes into the biorefinery. Secondly, there's also lignin, which is being considered, and instead of burning it to produce just energy, there may be some other beneficial uses. Today's talk is going to primarily focus on the process water and how we can beneficially use this waste. So I would like to now discuss the microbial electrolysis technology and what advancements ORNL has made in this field, and also then go to discuss the applications and evaluate how we can use this technology in the biorefinery, and then followed by going to the oil and gas industry. So the microbial electrolysis is shown here in this schematic. In this process, essentially, the wastewater that contains a variety of these compounds is fed into an anode, which is essentially a porous electrode, where it gets converted and broken down into unharmful products such as carbon dioxide, protons, and electrons. The protons and electrons are then combined in the cathode side to be able to produce a variety of products. If your goal is to produce electricity, you can use an aerobic cathode which produces water with oxygen, combining with electrons and protons, and which gives you an electricity. In case of if you want to generate hydrogen, you can do that by operating this cathode in a different way, where you apply an additional voltage to generate the hydrogen. So these are the two primary modes of bioelectrochemical systems: microbial fuel cells and microbial electrolysis cells, which have been investigated, and I'll be discussing what advancements ORNL has made in this field. Now, it focuses on production of and optimization of electrogenic organisms or biofilms, which are needed to efficiently recover the energy from these waste products. And that is done using nanowires, which are generated by these microorganisms, which are basically novel connective appendages which run from the microbes to the electrodes to be able to transfer the electrons efficiently. In addition to the operation of a bioelectrochemical system as a microbial fuel cell or electrolysis cell, there are a number of other products that can be also generated. For example, in this table, as it is shown, you can produce ethanol, for example, from acetate. You can produce even other chemicals, such as hydrogen peroxide, as well as other biofuels. Now, in all of these cases, one of the things that is required is an efficient anode, and that requires a high current production. So the proposed path for enabling that is to take the organics in the wastewater to be fed into the anode, which will be then converted to protons and electrons, and that can be converted to hydrogen in a cathode. So this slide outlines the intellectual property and the competitive advantages the ORNL technology offers. So we have 3 patents. The first one deals with improvement of the bioanode. The second one is on biorefinery application. And the third one is an application in the oil and gas industry. So in case of the bioanode improvement, what has been done is to improve the current density to achieve rates at which you can think about commercializing this technology. So I will discuss how we have done that in the next few slides. So improvement of the anode requires optimization of the electroactive biofilms. This requires consideration of 3 different kind of parameters: process parameters, system design parameters, and biological parameters. And there are a number of things that we have looked at and focused on trying to identify this space here, which will provide the optimal conditions to be able to apply this and produce high current densities at high coulombic efficiencies. So this has been done by combining state-of-the-art existing knowledge and built upon over the decade of research that has gone on in this area. And the focus has been on integrating process parameters, system design parameters, and biology parameters. So this has resulted in improvement of the current density to a level of about 35 amps per meter square. As shown in this result here, it takes about a month to achieve effective biofilm growth. However, it can be stably maintained at that current for a long period of time, of several months to years. And this has been done at a high coulombic efficiency of up to 80%. So now that we have developed the current densities to a level which are suitable for commercial application, I would like to discuss how this can be used and the variety of applications this is suitable for. The first one will be biorefinery wastewater treatment. In addition to that application, this technology is also suitable for application to treatment of municipal wastewater, food industry wastewater, as well as produce water from oil and gas industry. So I will be discussing the biorefinery application in detail in the next few slides, and at the end I will touch upon briefly on the produced water treatment application. So in the biorefinery application, we will look at the conversion efficiency of biomass and how that can be improved. So the main questions to be addressed in the biorefinery include conversion of the inhibitory compounds, as I showed in some of the previous slides, to electrons followed by hydrogen production. Develop a catalyst to convert not just a few, but essentially all of the organics, which is a variety of compounds from organic acids to phenolics to furfural, as well as unconverted sugars and organic acids. It's also necessary to show that the concentrations present in the biorefinery process water can be handled in the microbial bioanode, as well as to be able to recycle the water, it is necessary to completely remove these molecules and show efficient conversion at high current densities. Lastly, it is important to be able to demonstrate the application with real feedstocks. So we're going to start with a real feedstock, and in this case we used corn store to ethanol conversion process. And we used the sample that was generated from pretreatment of corn store followed by hydrolysis and fermentation, and we generated the biorefinery process water, which was then studied for conversion to electricity and hydrogen in a bioelectrochemical system. So this slide shows the effect of concentration on the current density and the coulombic efficiency. So what was done was, with 2 different model compounds, furfural and acetate, we looked at the representative concentrations, which can go as high as 2 grams per liter for furfural and about 10 grams per liter for acetate. And we demonstrated that even at the high concentrations, our performance of the anode is not affected. In other words, the anode is efficient enough and is strong enough to be able to handle high concentrations that are present in the biorefinery process water. In addition, conversion of the phenolic compounds was also demonstrated. Now, going from model substrates to real wastewater, we use as I showed in the earlier slide, the process water that was derived from corn store, and we demonstrated conversion of furfural, phenolics, acetate, and other compounds almost to negligible, undetectable concentrations in a period of 24 to 48 hours. When we go to application of this in the biorefinery, typically it would be a continuous operation. So we have done experiments to demonstrate that under continuous flow conditions, also you can do this process and be able to generate high current densities. So having demonstrated in the laboratory that this can be done, I would like to consider and discuss what are the implications of this for the biorefinery. As we started with this diagram, which was integration of MEC into the biorefinery, I would like to demonstrate where actually is the point of application for microbial electrolysis cells in the biorefinery process flow diagram. So the wastewater that is generated after fuel separation contains lignin as well as soluble organics. So we would include a solid liquid separation step to separate the wastewater and potentially find other uses for the lignin. The wastewater can then be fed into a microbial electrolysis cell to produce hydrogen or a fuel cell to produce electricity. We have done some estimates to figure out how much electricity or hydrogen can be produced in this particular scenario. So our calculations show that you can generate up to 2.5 megawatt in a full-scale biorefinery operating and producing about 70 million gallons of ethanol per year. Alternately, if the system is operated as a microbial electrolysis cells, you can generate up to 7,000 meter cube per hour of hydrogen. The energy produced is potentially sufficient to meet about 25% of the power needs of a biorefinery. So this is the added advantage of which can potentially reduce use of external electricity or fossil fuels, as well as produce a valuable product such as hydrogen, which can be used in the biorefinery. Now, this flow sheet that we have been discussing is primarily focuses on production of ethanol as the product. The future biorefineries are going to have complex combination of most efficient way of producing products starting with biomass. So I would like to consider a few mature biorefinery scenarios. There was a scheme that was provided in some work done by Lee Lin's group in production of various fuels and energy products from biomass. In this case, as you can see, the biomass is pre-treated with ammonia, followed by a fermentation process to make ethanol. The solid residue, which primarily contains lignin, was then gasified to produce a syngas and ultimately hydrogen to be able to fed to a Fischer-Tropsch synthesis to make hydrocarbon type of products. In order to do this, you require hydrogen. That hydrogen was generated from the waste stream through anaerobic digester process, followed by partial oxidation and use of cleaning steps to make the hydrogen available for Fischer-Tropsch. This kind of scheme was modeled by Laser et al., and they demonstrated the overall process efficiency to be about 70.4%. So this shows you that it is possible to go from the 53.5% for just ethanol production to a level as much as 70%. So we would like to consider this kind of advanced biorefinery scenario and see what kind of role microbial electrolysis can play in such a scheme. As I mentioned, there is a need for hydrogen for making fuels. And so the microbial electrolysis process fits in quite well to treat, which can be used to treat the wastewater and produce hydrogen directly, which can be fed into the Fischer-Tropsch synthesis process. Now, this eliminates the need for an anaerobic digester or partial oxidation and maybe even cleanup steps. So this has potential to be able to reduce the capital cost for the process, at the same time achieving high conversion efficiencies of about 70%. Now, this is a pretty relatively new technology, and there are several challenges. We have demonstrated that at the lab scale, we can achieve the performance of the current densities required for commercial consideration. The next step is to be able to see if this can be done at larger scale. And so the future directions that we are planning to pursue include understanding the scale-up process, using electrochemical methods to understand the effect of different process parameters on scale-up, and demonstrating ability to produce hydrogen at a current density of 20 amps or higher per meter square required for commercial consideration. Ultimately, what matters in the biorefinery is to be able to meet the goals that have been set up by EPA to be able to meet the renewable fuel standards. So there is a need to be able to demonstrate generation of 16 billion gallons of advanced biofuels or drop-in fuels by 2022, and this hinges on a main requirement for reduction in greenhouse gas emissions by 50% or more. This can be made possible by utilizing some methods which are able to produce more energy from the biomass, as well as some of the treatment cost for wastewater. This potential to be able to reduce these from 34 cents per gallon to lower. All of these goals can potentially be met by introducing microbial electrolysis cells and other bioelectrochemical systems in the biorefinery, which actually use very efficient processes such as biocatalytic and electrocatalytic processes to be able to achieve the high energy efficiency in the process. So with that, I would like to move on to a last application, which a couple of slides with focus on the oil and gas industry. As you all know, there has been a big boom for gas production in the US, and this has been made possible by the process called fracking or hydraulic fracturing. However, hydraulic fracturing also results in production of significant amount of wastewater. This is because large amounts of water are required to be pumped to be able to recover the oil and gas. So the issues that are related to the water that is produced from this process revolve around high organic concentrations or high BOD in the flowback water primarily, resulting from fracking, and high concentrations of salts present in the produced water as well as flowback water. Currently, there is a lot of involvement of trucking and shipping of the wastewater from these remote sites to water treatment centers, and this costs quite a bit, on the order of about $2 to $15 per barrel of wastewater. Additionally, there is a significant energy need or in some of the processes that are used to treat the waste on site. So we're considering how can the microbial electrolysis cells be employed in this particular scenario. So our goal in this particular development of this particular technology is to demonstrate a synergistic conversion of the organic contaminants into energy and using that energy to remove the salts. So this has been done by using a combination of two unit operations. One is called capacity deionization to remove the salts, and microbial fuel cells or electrolysis cells to convert the organic waste into energy, which is then used to run the capacity deionization. So we have demonstrated ability to generate energy from some of the waste, organic waste that is present in the fracking wastewater or the produced water in the presence of the high salt concentrations. Now this can be integrated with capacity deionization or salt removal process as shown in this particular diagram. And it is possible to remove at least some part of the salt using the energy produced by microbial fuel cells. However, the ability to be able to generate electricity from waste offers another option to run these electrosorption or capacity deionization systems by using waste from local sites to run this system. And so it may be able to reduce the cost of wastewater treatment as well as the energy intensive nature of desalination. So with that, I would like to acknowledge the funding for the work that I presented today and for the developments from EERE, Bioenergy Technology Office, as well as Bureau of Reclamation and the ORNL Lab Director's R&D program. One of the concepts that we are trying to pursue in all the work we have been pursuing is to combine the bioelectrocatalysis using microbial fuel cells, electrolysis cells, with the advances in the biorefinery for biofuel production so that you can have a overall increase in energy efficiency in the biorefinery. So with that, I would like to stop and take any questions.
Thank you, Dr. Baroli. We now will ask a few questions that our viewers have sent in during the webinar. One question that was sent in was, for the technology to be adopted commercially, what is the next step for scaling up?
So as I mentioned, we have demonstrated the ability to be able to produce current densities on the order of 35 amps per meter square at the laboratory scale. It's very important to be able to achieve these kind of performance at the higher scale. So our next step is to look at scale up and experiment with systems of about 10 liters or so to be able to demonstrate and identify the scale up parameters to go to the next stage.
Thank you. We have another question. It is, what is the lifetime of the biocatalyst?
So the biocatalyst that is used in the anode is a living organism. And so it has the ability to be able to reproduce. And so the lifetime of the biocatalyst is essentially infinite. And it's possible to be able to regenerate and grow the catalyst for several years.
And we have time for one final question. The question is, what are the competing technologies for this system?
That's a great question. So as I demonstrated in some of the slides, the mature biorefinery scenarios have used a wastewater treatment option to be able to recover more energy from the waste organics in the wastewater. And anaerobic digester was one of the technologies that was used. So that's a competing technology which is currently available and it's commercially viable to be able to convert and produce energy. However, the anaerobic digestion process produces a methane gas which then requires further processing to be able to use in the biorefinery, for example, for upgrading of fuels and so on. And so the alternative that we have here, which produces hydrogen directly, can be a much more favored process and potentially reduce costs also for the biorefinery.
Thank you.